Hi everybody, and welcome to this video on patterns of inheritance from the Science 10 Genetics unit. Before you watch this video, you need a copy of some fill-in-the-blank notes that you can get in the classroom or you can find on Teams. So get those now, get something to write with, and then come back. All right, let's get into it. So just a quick review of some things that we've been going over, but linking grade 9 to grade 10 science. So different forms of the same gene, remember, are called alleles, and organisms possess a pair of alleles for each heritable trait. Heritable means it can be passed from parent to offspring. So these are things that are coded for in your genes, not things that you learn. So some traits have just two alleles. So for example, having hair on the middle joint of your finger, or having a hitchhiker's thumb versus a straight thumb are controlled by just two alleles in humans. So either straight thumb or hitchhiker's thumb. Some traits like blood types have many different alleles. So you can have A, you can have B, you can have O type blood. So there's more than two options. Some traits are controlled by more than one gene. This is called polygenetic inheritance. So stuff like our height, our skin color, our eye color, our weight are controlled by lots of genes, on sometimes even on different chromosomes. But we're going to make it simple for these first few kinds of problems. We're going to talk about Mendelian traits, and they're named after an Austrian monk named Gregor Mendel. He was the first to experiment with inherited characteristics, and he didn't even know anything about DNA, which is pretty amazing. So we know now, being able to look into cells, we know that in meiosis, pairs of alleles separate. So during fertilization, those gametes then recombine when the sperm and the egg fuse, giving us one allele of each gene from each parent. And we have two alleles of each gene total. So you're writing down anything in this slideshow that's underlined. So write down two alleles of each gene total. Each sperm and egg, or each gamete, will carry a different set of genes, which explains why siblings look similar but not exactly the same, not identical. In traits that are controlled by one gene, one allele can be dominant to another, and the non-dominant trait is called recessive. And what this means is that if you have one copy of the dominant allele, it overpowers the recessive one, and you will have whatever that trait is. So let's look at an example. Here we have a mother and father horse, and you can see the mother's gametes here. These letters stand for alleles. And when you see a capital letter means that this is dominant. So it says in horses, the allele for black hair is big B. That means it's dominant to the allele for red hair, which would be this little b. So this one's recessive. It looks like the father in this situation has two recessive alleles, which means that the father has red hair. The mother has a copy of this dominant black hair allele, so that means that she has black hair. One copy of the dominant allele is enough to make that the trait that you see. So if you have this mother and this father, what are the chances of a black-haired mother and a red-hair father having offspring with red hair? So let's look. How many of these offspring have red hair? Not this one, because it has one copy of the big B, black. Not this one. So this one and this one have red hair. So our probability is a 1 in 2 chance, or you can also express it as a percentage and say it's a 50% chance. So we're going to talk a little bit about probability in a couple slides. Okay, some more words that you're going to need to keep coming back to, so make sure you have these copied down. Genotype means the alleles a person contains. So the genotype of those horses, for example, the mother was big B, little b. Phenotype means what genes are expressed. So this is what a person looks like, or what traits an organism has. So a phenotype would be black hair or red hair. You can also be homozygous. So that's an organism with two of the same alleles for a particular trait. Or heterozygous, which is an organism with two different alleles for a trait. So I'm going to show you some examples. So in any situation, you only have three options for genotypes. So you can be homozygous dominant. So the word homo means same. So for example, you probably hear it most common in homosexual is someone who's attracted to the same sex as themselves. So homozygous dominant means two dominant alleles. I forgot to underline these. I'm sorry. Please copy these down on your notes. You can also be homozygous recessive, which means you have two recessive alleles. Or you can be heterozygous, which means one dominant allele and one recessive allele, one of each. Hetero means different. So like heterosexual is someone who's attracted to someone of the opposite sex to them. Okay, so two dominant alleles would look like, let's keep using B, big B, big B. Write that down as an example. Homozygous recessive would be two of the little letters, so little b, little b. And heterozygous would be one dominant allele and one recessive allele, so big B, little b. Now, in the type of inheritance we're talking about now, the heterozygous one has the same phenotype as the homozygous dominant one. So both of these, in our horse example, would have black hair. Oh, it's hard to write using a mouse. And this one would have red hair. That's the phenotype. Okay, let's do some more practice. In humans, you can have a free earlobe, which you can see down here, means it's not attached to the side of your head. Or you can have an attached earlobe, so it's stuck to the side of your head. And free earlobe is dominant to attached earlobe. So if your genotype is big E, what kind of earlobes would you have? You'd have free. Your genotype is free earlobes. Okay? If you have little e, you are going to have these recessive earlobes. Your phenotype is attached. And if you have big E, little e, so you're the heterozygous genotype, your phenotype is going to be the dominant one, so free earlobes. Do you know what kind of earlobes you have? Pause the video and look in the mirror or use your selfie camera. So we can learn a lot about the genes of parents or offspring by doing something called a genetic cross. A genetic cross is a deliberate mating between a genetic male and a genetic female. So you're choosing the parents carefully in a genetic cross. Whenever you see this little X, it means crossed with. So this is big R crossed with little r. A monohybrid cross looks at just one trait. So for example, just the hair color on the horse, or just the earlobe, versus something that uses more than one, looks at more than one trait. Hybrid is any offspring that has different traits from its parents. So if your offspring has different traits from the parents, we call it a hybrid. Okay? And we have a really helpful tool here called a Punnett square. It helps us determine the probability of offspring inheriting certain traits from their parents. So it shows the genotypes of the parents and the offspring. And I'm going to show you how to set one up. But basically, you look at your male and you put one of its alleles above each box. You look at your female, you put one of its alleles above beside each box. It actually could be male here and female at the top. It doesn't really matter. But then for your offspring, you bring these genes over. Okay? Bringing the genes down and this one over to see what your combinations are. After you do a Punnett square, you might be asked for a phenotypic ratio or a genotypic ratio. The phenotypic ratio shows the frequency of phenotypes or the traits you see in an offspring. So for example, maybe in a Punnett square, 3 of the boxes have purple flowers, 1 of the boxes has white flowers, so your ratio is 3 to 1. The genotypic ratio shows what your genotype is. So you could have, for example, 1 big B, 2 big B little b's, and 1 little b little b. Just a reminder on how to calculate probability. Hopefully you've covered this in math class, but the probability is the number of chances of an event over divided by the number of possible events. Okay, so for example, if you have a 1 in 4 chance of inheriting a recessive attached earlobe out of 4 boxes, you're always going to have 4 options in a Punnett square, so your ratios should always be like 1 over 4, 2 over 4, or 3 over 4, 4 to 4. So keep that in mind. You can give it in a ratio, which looks like that, a decimal, a percentage. You could also give it as a fraction. I should add that fraction. Okay. A way of doing this without listing all the different options is called the product rule. So consecutive events can be calculated using the product rule. The probability of independent events happening at the same time is the product, which you get from multiplication, of the probability of each event. So the probability of the first child being XY is 1 in 2. The probability of the second child being XY is 1 in 2. But the probability of having 2 XYs in a row, we're going to multiply. So 1 over 2 times 1 over 2. Do you remember your fraction laws? It's 1 over 4, or 25%, right? You multiply the top by the top, the bottom by the bottom. Okay, we're going to do an example of a Punnett square together now. So in humans, freckles, big F, are dominant to not having freckles. So what are the probabilities for each genotype and each phenotype? So let's say we have a mother with a big F, little f. What does she have? Does she have freckles? She does, because she has 1 copy of the dominant allele. Our father also has freckles, but has only 1 dominant allele and 1 recessive. So what we do here is we bring this top one down and bring the one from the left over. So this offspring has 2 big F's. Here we also bring them over. It's convention in science to write the capital letter first. I'm trying to make my little f's really curly so you can tell it's little. I'm going to add a little tail. Okay. This one also has a big F and a little f. So we write big F and then little f. And this one has 2 little f's. Forgot my tail. Okay, so it's homozygous recessive. So what are the probabilities for each genotype? Let's write a ratio. So here we have a homozygous dominant, big F, big F. We have 1 of those. Okay, so 1 ratio has these 2 little dots. Okay, and then we have 2 big F, little f's. These are both heterozygotes. Oh no, that's a little f. Okay. And we have 1 of the homozygous recessive, little f, little f. Okay, that's our genotype ratio. If we're doing phenotype, let me get a different color. Because this is a dominant recessive relationship, we have a dominant allele, all three of these ones I've circled in green actually have the dominant phenotype, which is freckles. So we have 3 freckles, 2, 1, no freckles. That's our phenotype ratio. I hope that was helpful. Sorry this one's a bit long. Have an excellent day. You can go do your practice now and let me know if you have any questions. Bye.